I'm going to talk a little bit on our efforts in breeding for resistance and tolerance to zebra chip using sort of going outside the normal traditional potato and looking at some of the wild species as additional sources of resistance as well. I'd like to begin by acknowledging I couldn't do this by myself, and this is where the efforts of John, Trumble, Sean, Beatrice, and Joe to help in identifying and characterizing a lot of the germplasm we have to see what could be useful for ZC tolerance or resistance. I'm going to begin by talking, give a little background on what do we know about resistance to ZC in potato varieties. And a very nice study was done by Joe in 2011. Where he had field cage evaluations conducted, 9 varieties, most of which are widely grown in North America. You can see them listed there. 2 years of evaluations, and Joe had some very nice quotes in there, and these are 2 of them. So one was potato yield in all the tested varieties were impacted by ZC, with yield losses ranging from about 50% up to 87%. All potato varieties evaluated, he considered susceptible to the disease, with almost 100% developing severe ZC foliar and tuber symptoms. A second nice study was done about 2 years later by John Anderson. He's a potato breeder there in New Zealand. Obviously, we have a New Zealand component here as well. And a very similar approach where they were looking at resistance among some of the more widely grown potato cultivars in New Zealand. And what they found, they had field evaluations conducted in 2 years, and they actually had a full, reduced, and nil insecticide applications. And I was really impressed by the full. That's a lot of insecticide applications, where a pre-plant and 13 to 15 foliar applications during the year, and that was what they considered necessary for control of ZC. And what they found, they looked at 12 varieties grown in New Zealand, and you can again see Ranger Russet and Russet Burbank, which were 2 of the ones grown in North America too, very widely grown in the US and Canada. Now, they did find that Nadine, which is a fresh market, had very little ZC symptoms in raw tubers, sort of similar to what Joe was indicating with some of the ones he's seen in his evaluations. But when they looked at the difference in yield between full and nil treatments, even though they didn't show the tuber symptoms, very dramatic cut in yield. And so similar to what Joe had observed, they had a quote too, where there's real no indication that any of the other cultivars in the trial had effective resistance to tomato potato psyllid or ZC disease. So those were some of the better studies that were conducted to look at ZC resistance in widely grown varieties in North America as well as in New Zealand. So the question is, there doesn't appear to be at least, there's 4,500 potato cultivars out there, and this is a small sampling, but nonetheless they're the most widely grown. So my thinking was to look outside when we run into issues like this, look outside the normal gene pool of cultivated potato, and let's look at some of the species relatives. And in our program, we have been working with one, this is the first category I'm talking about. It's a tri-species germplasm. It has a species relative of cultivated potato from Chile. It's non-tuber bearing, and it was brought into the program. It had some very nice virus resistances as well as insect resistances. You can see green peach aphid. That's a vector of many of these viruses. And the other component of this tri-species germplasm was tuberosum, so your normal cultivated potato, but then Bertholdtii. And that's of Bolivian origin. It's tuber-bearing. The PI is a plant introduction. This comes from NRSP6, the gene bank in Sturgeon Bay, Wisconsin. And these were some of the resistances that were categorized for this PI. these two clones were somatically hybridized as part of actually my graduate student dissertation. And I've sort of followed it through since that background because it's sort of unique material. And in the progeny, the somatic hybrids, we were able to get progeny from those. And the progeny showed green peach aphid resistance. So you had reduced fecundity, growth inhibition, nymphal survival. And this is done with Dave Ragsdale and Ted Radcliffe. And so it was a very nice study showing the species did confer resistance to green peach aphid. Colorado potato beetle, this material also showed reduced field defoliation. Wireworm, this was done with Juan Alvarez when he was in Aberdeen, and showed reduction in tuber damage and comparable or better than mocap controls as well. So it seemed like good material to look at for resistance to psyllid, and that's why this material was utilized in our screenings. And this is where it's worked out quite well in our collaboration with John Trumble and his personnel in his lab. And this is a published paper with John and Casey Butler, who was a graduate student at the time. They looked at behavioral bioassays and LS transmission assays in their lab there on this tri-species material. Looked at 4 generations. So this is 4 generations from the somatic hybrids, each one incrementally improving in terms of their agronomics. 3 and 9 had significantly reduced feeding occurrences, and 4 and 9 had reduced resting duration. So this material, and this is across the backcross generations, not just the first backcross. So what that was indicating was there appeared to be some impact on psyllid behavior in this material. And another aspect that Casey found was that there was reduced LSO transmission in 3 clonal generations. Psyllid resistance may be contributing to that, but one of those 3 actually had no differences in psyllid behavior relative to Atlantic, and yet it also showed a reduced LSO transmission. So that bode well for possible even resistance to LSO itself, not just the insect vector. Another nice study was conducted just this year. It was published with John Diaz-Montano, and that is looking at anti-zoonosis and no choice on 2, a backcross 1 and a backcross 4 progeny from these tri-species germplasm. There were no differences for number of adults or eggs laid among entries in a 24-hour period, so it didn't appear to be anti-zoonosis involved. With no choice assays, though, there was a difference. There was no difference in days for egg and nymphal instars, so development was not impacted. But when they looked at the number of N5 in adults relative to Atlantic, there were differences that were observed. So that would indicate there was some antibiosis potentially contributing to what was observed in those studies. I'm going to talk, Joe's talked about it, and also Doug today, and this is that material that looks promising. And this is actually material from Shelley Jansky's program. She's an ARS colleague of mine in Madison, Wisconsin. I brought it into my program in 2007 for its cold sweetening resistance to use as a parent. When I became involved with the ZC project, I decided that might be good material to include in Joe's initial screenings there. And the reason for that is it has a varied species background there. You can see 3 different species in its background. So as Joe mentioned, it's had over 4 years of field evaluations by him with none to light symptoms and cut tumors. And he's also done chip evaluations with no to little ZC. And Sean had looked at this in 2013, Prager, with John Trumble, and it appeared that anti-zoonosis, where you had increased psyllid jumping with reduced duration of resting, might also be involved with this material. And the last one I'm going to talk about in terms of the germplasm discussed in this presentation, this is PALB. There are 2 clones, -3 and 6. It's a Chuck Brown cross where we selected at Aberdeen, Idaho, and it's been maintained in our program in Aberdeen. And the background on that is it actually has Gemstar as one parent and this LB531. And that is Guerreroense is the background on that. It's a late blight resistant species out of Mexico. And the progeny of this material was identified as having putative ZC resistance in 2011 in Hermiston, Oregon. And first standardized evaluation, Sean looked at it last year, again similar to the previous clone. There might be anti-zoonosis involved, and neither clone displayed LSO infection in Sean's 2013 transmission assay. Albeit we probably need to look at some of the frozen samples yet, but the question arises, does it have resistance then to LSO? So we're moving now to some of Sean and John's work this year with No Choice Bioassays. Some of the background, you can read the protocol there. And basically 3 male-female pairs, cage, you had two cages per plant. And psyllids removed following 72 hours. And the leaflet with cages were marked and plants retained for 21 days. And with respect to eggs being laid in that 3-day period there, what was interesting, here's Atlantic as your control. This -211 actually had a greater number of eggs laid, whereas some of these other clones, this is the only one that had statistical significance, and that is a backcross 2 of that tri-species material we had talked about. So there you can see there was, even though these others aren't statistically significant, there does appear to be a biologically significant reduction there. So that was good to see that there may be some antibiosis again showing in this material. The growth index, this is talked about by Sean yesterday. And this is just showing again, you have 2 individuals here. So you can see where there's statistically significant reduction in the growth index relative to Atlantic. This other one also appeared to be reduced, but I talked to Sean about that, and there was an outlier that sort of skewed, and it didn't allow for significance there. But if you remove it, then this individual would be considered resistant as well. But, you know, that's part of the deal. It's got to be included in there. Sean also conducted 2 choice bioassays with Beatrice this year in 2014. You can sort of see here the layout where a leaf from Atlantic and a leaf from the other entry are put inside this plastic. And then you have male-female pairs put in. And you can see the protocol again. And there they found, and this would be for trying to look for anti-zoonosis or something that impacts feeding behavior or repellency to the material. And they found no significant differences over a 4-day period there, though. This is some results from last year with, again, in Trumble's lab. And you can see this is QPCR results. And what was interesting to me, as I indicated, were the PALB clones from the No Choice. You can see this is your control Atlantic coming across at about 50% infection. And then you had individuals in that No Choice that had no, with QPCR showed no LSO. So that was very exciting. It was even a little more exciting where at least 2 of these had previously been identified by a Butler as having reduced LSO infection rates. This other one, however, as you can see, Butler had shown as well that it was significantly reduced relative to Atlantic, but this time around you can see it wasn't. But at least we had 2 of the 4 sort of being repeated over time there. Moving on, I'd just like to say I'm enjoying also working with Arash Rashed. He's at Aberdeen, and he's been a great colleague to have there. He's very interested in a lot of this work, and he's been doing, we provided him with 11 breeding clones, and he's been looking at transmission studies with these LS clones in the greenhouse. It's going on right now, actually. And so this is sort of his screening protocol. And with these clip-caged LSOB infected psyllids then, He's sampling them over time, and we're going to see how LSO is impacted over time. He's done 3 leaf samples already. We'll probably have about 2 more in November and then harvest the end of December. So I'm really interested to see on the transmission side, too, what Rosh's data shows. So some of you are probably wondering, well, that's great. We've identified maybe putative, well, we know for sure we have psyllid-resistant material, might have putative tolerant resistance to ZC. How do you go about moving that into something that's useful for industry? And as it was alluded to by Doug, hybridizations are conducted with this resistant material in the greenhouse with the flower, directed hybridizations among resistant as well as other parents that are better adapted perhaps than this material. If successful, then you have berry production, just like a small tomato. These are potato fruit. And within them are what are called true potato seed, and that's the starting point for our breeding program. And that true potato seed is germinated like you would any seed from sexually hybridized crops, and put into individual pots in the greenhouse, and we harvest the largest seedling tuber is what we call it. So each of these individuals in that little flat have the same maternal-paternal parents, but just like your siblings, each one is different and has the potential to be a new variety. So these seedling tubers are packaged, put in the cooler over the winter, planted at 2.5-foot spacings, and that's the first field year. From that point forward, they're all going to be asexually propagated. Lovely thing about potato. So we work at both the sexual reproduction as well as asexual. And then over time, if those are selected, we look at them in replicated trials over years, over locations. And this is some of the material coming forth, which has been indicated as having either psyllid resistance or ZC resistance. So we have material that has the round shape desired for the chip market class. We also have material that has the long russet type as well, coming from the tri-species hybrid material. And just to give you a summary here, this is in 2014, the 3 sources I've talked about in this presentation. These are the number of first field selections. So these are in the field the first year that were made from hybrids using this material. And this is material where we have them in more advanced trials, replicated trials, and we'll continue with them into 2015. So to summarize, species germplasm can be a very good source when you're having difficulty finding resistance in cultivated material. It does take some work, but it's worth the effort. Some background, the E. tuberosa bertholdtii, it's been shown to have psyllid resistance and shown to also and documented to have anabiosis relative to Atlantic. And we also have the M5 from Shelley Jansteed's program. Also looks like it's been very good, as Joe indicated in his trials over several years, as well as Doug. So it's exciting. I think there is some resistance tolerance we can work with in this material. And then the Guerrerense germplasm. And this one I'd like to do more with the LSO transmission, because I think we could have some possible resistance coming from this material as well. And then lastly, if I can get it to work here, there we go. We have had success in using this material, and we're moving ahead with developing cultivated market classes that have more impact for the industry, obviously. But yeah. I'm excited by the collaborations we've had in this project, and I think it's been, I'm very appreciative of the collaboration with the entomologists and pathologists I've worked with. Can't do it by myself. And with that, I'd just like to thank you for your attention.